Hello students, today we will study about the structure of proteins. The structure of proteins are of four types: primary structure, secondary structure, tertiary structure and quaternary structure. The primary structure of a protein is determined by its amino acid sequence. The term secondary, tertiary and quaternary structures are frequently applied to the configuration of the peptide chain of a protein. The secondary structure is determined by the spatial arrangement of the main peptide chain. Spatial arrangement, that is the arrangement in the three dimensions. The tertiary structure is determined by both the side chains and other adjacent segments of the main chain. Side chains and different type of segments which are near about. Finally, the term quaternary structure is used for the arrangement of identical or different subunits of a large protein in which each subunit is a separate peptide chain. So conclusively we can say that the primary structure is the result of amino acid binding. These are bound by peptide bond. So amino acid sequence is result of the primary structure. And the term secondary and tertiary and quaternary structures are due to the configuration of the peptide chain. And now we have a short look for the different structures and what is the configuration of the different polypeptide chains in the protein. This is the primary structure of the protein. In the primary structure, these small balls or small beads are for the amino acid, different amino acids which are joined together by the peptide bond. And so we can say that the different amino acids are assembled together by a peptide bond, by different peptide bonds or many peptide bonds to get a primary structure of the protein or a polypeptide chain. Now these polypeptide chain are folded and they result in the secondary structure of the protein. This secondary structure is of two types, alpha helix and beta pleated sheet. Alpha helix is the helical form and in the beta pleated sheet these are like the sheet form. And these are the result of the hydrogen bonding interactions. So these are the result of the folding of the polypeptide chain. The third structure is the tertiary structure and this is the result of packing of beta pleated sheet and alpha helix. The fourth structure is the quaternary structure and this is the result of the interaction between the same polypeptide chain and different adjacent polypeptide chains. Now we come to the primary structure. There are 20 different standard L type of amino acids used by the cells for the protein's constructions. Students, we have discussed earlier that alpha amino acids are of have the two type of configurations D and L. But most of the naturally occurring alpha amino acids are of L configuration. So we are saying here that there are 20 different standard L alpha amino acids used by the cells for the protein constructions. Amino acids, as their name indicates, amino, that is amino group, and acid means carboxyl group. So amino acid, as their name indicates, contain both a basic amino group, that is NH2 group, and an acidic carboxyl group or carboxylic group, that is called as C double bond O OH group. This difunctionality means having amino group and carboxyl group together. So this difunctionality allows the individual amino acid to join in long chains by forming peptide bonds, because they are joined in the by the polypeptide bonds. So these are the long chain is formed. Amide bonds between the NH2 of one amino acid and carboxyl of another. What is the result? When amino group of one amino acid and the NH2 group of one amino acid and the carboxyl group of the another amino acid join together, there is a loss of water molecule. There is the loss of a water molecule and the formation of CuNH bond, which is called as peptide bond. Sequences with fewer than 50 amino acids are generally referred to as peptides. It is saying that 50 amino acid or lesser than 50 amino acids, when they join together, then they are simply called as peptides. While the terms protein and polypeptide, this term is used for the amino acid when these are more than 50 or many more in counts. So while the terms protein and polypeptide are used for the longer sequences means which are having more than 50 or larger number of amino acids. A protein can be made up of one or more polypeptide molecule. Okay, protein is the protein is the result of the binding of one or more polypeptide molecules. The end of the peptide or protein sequence with a free carboxyl group is called the carboxy terminus or carbon terminus. It is saying here that the peptide or protein peptide or polypeptide chain which have the one end with the carboxyl terminus, that is COOH terminus, is known as C terminus, and the term form amino terminus. Amino terminus means the end have the polypeptide chain, its end is NH2. It ends with NH2 group. It is called as N terminus and describe the end of the sequence with a free alpha amino group. Look at here. In this diagram, it is shown easily that this is a polypeptide chain which is formed by the joining of amino acid, many amino acid which are joined by a peptide bond, that is CONH bond. And its terminus, this last amino acid or the amino acid which is in the last have the carboxyl group at the end, so it is known as carboxyl terminus or C terminus. And here its end is the last amino acid, its end group is the NH2 group, and this is called as N terminus or nitrogen terminus. These all are the small balls or small beads are the amino acids which are bound together by the peptide bond. The amino acid differ in structure by the substituent on their side chain. Each amino acid have the different substituent. It may be alkyl substituent, it may be aryl substituent, or it may be another type of substituent. These side chains confer different chemical, physical and structural properties because different groups are there. So their chemical, physical and structural properties will be different, and to the final peptide or protein. The structures of the 20 amino acid commonly found in proteins are shown in figure. We will see here. Each amino acid has both a 1-letter or 3-letter abbreviation. These abbreviations are commonly used to simplify the written sequence of a peptide or protein. Look at here in this diagram, we can easily see what it is saying. It is saying that it is a polypeptide chain in which each box is conferring one amino acid. This is one amino acid, lysine. So first 3 letters are abbreviated here. Lysine, Lys, lysine. This is for lysine, this is glycine, this is glycine, this is leucine, this is valine, this is alanine, and this is histidine. Similarly, all other amino acids are written as their first three letters in the abbreviated form. These are also called amino acid residues, and this results in the primary structure of the protein. Now we come to the secondary structure. The secondary structure is the result of the stretching or the folding of the polypeptide chain. Stretches or strands of the proteins or peptides have distinct characteristic local structural conformations or secondary structure dependent on the hydrogen bonding. It is saying that the secondary structure is the result of hydrogen bonding, and also it is the result of the different conformations which have been exhibited by the polypeptide chain by its folding. The two main types of secondary structure are there: the alpha helix form or alpha helical form and the beta pleated sheet form. Two types of secondary structure are there. The alpha helical form or the alpha helix is a right-handed coiled strand. It is a coiled structure like a coil, and the side chain substituents of the amino acid groups. Side chain substituents are there which may be alkyl or aryl or different groups in an alpha helix extend to the outside. They extend to the outside and they form the hydrophobic interaction or hydrophobic bonds. The hydrogen bonds is formed between the oxygen of each carbonyl bond in the strand and the hydrogen of each NH group, 4 amino acids below it in the helix. It is saying that the hydrogen interaction or hydrogen bonds are formed, but where it is formed? It is formed if the first amino, the carbonyl group of the first amino acid, let it be the first amino acid, its carbonyl oxygen is O, and after first the fourth or the fifth amino acid having NH group, and the hydrogen of this NH group is getting interacted with this oxygen, carbonyl oxygen of the first amino acid. These form the hydrogen bond. The hydrogen bonds make this structure specially stable, very stable, and the side chain substituents of the amino acid fit in beside the NH group, and these are substituents are get fitted in the NH groups. Look at here, what it wants to say is that here it is the alpha helix form. It is saying that the carbonyl oxygen of the first amino acid, this is the carbonyl oxygen of the first amino acid, and the NH group of the fourth amino acid, the hydrogen of the fourth amino acid, that get interacted with each other and form the hydrogen bond. Here this is the carbonyl group, and this is the first, this is the amino acid, and this is the second, third and fourth, and this is the fourth. Okay, so carbonyl oxygen of the first amino acid and the hydrogen of NH of N attached to N nitrogen, this is the fourth to the next amino acid. Okay, that is this bond is formed from 1 to 5, between 1 to 5 amino acid, and these, due to the result of this hydrogen bonding, there is a alpha helical form or coiled form is formed. The next is beta sheet structure or beta pleated sheet. The hydrogen bonding in a beta sheet is between the strands. That is interstrand. That is when two strands are there, two polypeptide chain are there, then the hydrogen bonding occurs between the two strands, two polypeptide chains. Then beta sheet is formed rather than within the strand. When within the strand hydrogen bonding is formed, hydrogen bonds are produced, then the alpha helix are produced. So the alpha helical structure is produced when or is formed when the hydrogen bond is within the or the intrastrand or in the single strand hydrogen bond is there. And when the hydrogen bond is formed between the two strands, then beta sheet is formed. The sheet conformation consists of pairs of strands lying side by side. When two strands lie side by side, then the sheet is formed. The carbonyl oxygens in one strand bonds with the amino hydrogens of the adjacent strand. How this is formed? Look at here. This is the one strand, one polypeptide chain, and this is the another polypeptide chain. Okay, this is the first strand and this is the second strand, and the carbonyl oxygen of the first strand is interacted with the hydrogen of the N of the second strand. Here the hydrogen bond is formed. This is the first strand and this is the second strand, and the hydrogen bond is getting formed or getting interacted with the both of these strands. So it is the interstrand hydrogen bonding, and this is the result of intrastrand hydrogen bonding. This is a single strand here and this results in the alpha helical form, and here the two strands are there and this results in the beta pleated sheet. So this beta sheet it may be parallel or anti-parallel. How? The sheet conformation consists of pairs of strands lying side by side, and the carbonyl oxygen in one strand bonds with the amino hydrogens of the adjacent strand. The two strands can be either parallel or anti-parallel, depending on whether the one strand directions is nitrogen terminus to carbon terminus are the same or opposite. That is, both of the ends have the same nitrogen terminus. Look at here. Both of the strand, either these may be carboxyl terminus or nitrogen terminus. These are the parallel, and it will be anti-parallel if the one of the strand have the end group nitrogen terminus and another strand have the end group with the carboxyl terminus. So these two strands may be parallel or anti-parallel depending on whether the strand directions are the same or opposite. The anti-parallel beta sheet is more stable due to more well-aligned hydrogen bonds. And this is reflected here that the anti-parallel beta sheet is more stable because there is the more aligned hydrogen bonding is there. These are the strands having different terminus here, ends here. So this is the anti-parallel, and this results in the more stable beta sheet here. So students, we conclude our lecture here. The next structures, tertiary and quaternary, will be explained in the next lecture. Thank you.